everybody, Mrs. Cooper here. We are on day 3 of DNA technology, and this will be our third lecture for DNA technology. We always begin with some warm-up questions as a refresher from last class, so let's read through those questions. What type of virus can perform transcription backwards? What type of molecule ends in ase? What is the difference between blunt and sticky ends? And what is the molecular glue that binds DNA back together? And let's check our answers. So first off, we've got the first question, what type of a virus can perform transcription backwards? So we're looking for a little virus, so we know virus is going to be in the name. Performing transcription backwards. This was when those viruses took their RNA and used an enzyme called reverse transcriptase, so hence the word backwards, to copy their RNA into DNA and incorporate it into the host cell. So the organisms that perform this are retroviruses. Another hint is that R for retrovirus, R for containing RNA as their genetic material. Our next question is, what type of molecule ends in ase? This one's pretty easy. I also believe it was a warrant question from last time, so hint, hint, it's something you should definitely know. Enzymes, they always end in ase. Helicase, ligase, DNA polymerase, reverse transcriptase, they all end in ASE, ACE. Our next question down, what is the difference between blunt and sticky ends? So ends, what is the end of? It's the end of DNA pieces that are cut by restriction enzymes. Blunt ends are flat, it's a cut straight through the DNA. Sticky ends are staggered, so we've got our DNA with a little single piece, another chunk of DNA with a single piece, and that these sticky ends match up to each other according to Chargaff's rule. So very important, those sticky ends. And last question, what is the molecular glue that binds DNA back together? So the glue that can bond those sticky ends together is DNA ligase. You could think of ligase as something that is linking DNA back together, Li, linking, ligase. So I always like to use word associations like that to help with, okay, how can we remember all of these new biological terms? Hope everyone is doing well today, and on our agenda for day 3 and lecture 3 is that I plan to talk about both stages 3 and 4 today. So at the same time, 2 stages in 1. And I did have an activity planned, but since we're not in the classroom, we don't have to do that activity, or we can do an abbreviated version of it. Stage 3 is titled Transformation. So for transformation, it reads, let's read along together. It says the recombinant DNA plasmid. So before we go any further, let's make sure we know what that means. Recombinant DNA plasmid, meaning we combined DNA from 2 different sources. So if you recall, a plasmid was a circular chunk, and, well, that eraser tool just doesn't work very well. A recombinant plasmid was a circular chunk of DNA that we had cut out a chunk and added a new piece in. So it is, we recombined 2 different things here. And that plasmid was found inside of a bacteria. So once we take that plasmid, we have changed it. We have added new DNA to it. We now want to put it into a new host cell. That will probably be another bacteria, because bacteria contain plasmids already. The process of putting that recombinant plasmid, look at that, I need more coffee, of putting that recombined plasmid, 2 different DNA sources, back into a bacteria, that process is called transformation. We're going to be transforming the bacteria. So the host bacteria and the altered plasmids get mixed together. So you kind of put them in a big bowl, mix them together. This has to be done at a high temperature. Let's think about that. If we have, let's say, a nice little bathtub here. We put that bacteria in it. Oh wait, I didn't want to do that part yet. We put the little bacteria in it, and we also put that plasmid chunk with its new little DNA. A nice, warm, steamy bath. So there's little heat marks there. It's really nice and warm. What do you feel like when you're in a bath? Do you feel warm, relaxed? Definitely. So the goal here, by altering the temperature, so by increasing the temperature, the bacteria's cell membrane around the outside helps hold the bacteria together like the bacteria's skin. It will become permeable. That was a word we learned back during the unit on cells. Permeable meant things can now go in and out through a membrane. So if that bacteria's membrane is permeable, it will allow things in and out because it is relaxed due to that high heat and that temperature increase. It means that the plasmid, this guy here, or girl, either way, this little plasmid can make its way into the bacteria. It can go through, it can permeate that membrane in the bacteria and get inside. Once that happens, like our picture over here, our recombinant plasmid, our host bacteria, once that goes inside right here, we now have a transformed cell or a hybrid cell. Hybrid meaning two sources. We have the two sources, the new DNA here. All right, I hope that that really helps to clear up what is transformation. We're transforming a host bacteria by adding new DNA to it, by using heat so that that plasmid can get inside. All righty. Stage 4. See how quick that was? Stage 3 is just one slide. So we move right back on in our notes to stage 4, which is identification. Now for identification, it's good to know which cells actually relaxed enough to let that plasmid inside. Only about 0.001%, that's a very, very, very tiny percent of bacteria actually accept or allow that new plasmid to go inside of them when they're mixed together. Because very few cells are going to transform, it's important to identify. We want to know exactly which bacteria now contain that recombinant DNA. There's two ways of identification. So again, we've got two ways of identifying something because you don't know what genes you put in there. Or well, a scientist is going to know what genes, but you don't know which bacteria actually have them until you try a couple of these different steps. And depending on the gene that you're inserting into the bacteria, some steps in the process of identification are going to be useful instead of others. So here we go. If you identify recombinant plasmids, you can use two types of gene markers as your identification tool. Remember, these are our two types of identification. A gene marker, which we're going to learn about in a second, is kind of like flags, showing you exactly where a gene is. I'm going to show you how to use fluorescent protein to do that. And we're also going to see antibiotic resistance. Recall we talked about plasmids containing these genes. That's the second way of a type of marker or identification tool we can use to see which plasmids have that new gene that we inserted. These gene markers can actually be attached to whatever new gene you're adding. So when you cut the chunk out of a plasmid and you go to put new DNA in, before you put that new DNA in, you can attach a gene marker to it. Something like a flag or antibiotic resistance that show you exactly that that cell has the new gene that you wanted. So it says if the gene marker, whether it be antibiotic resistance or fluorescent protein, if the gene marker is expressed, that's a genetics term, it means showing, actively producing whatever the gene says to make. then we know that that bacteria was transformed correctly, that that new gene was correctly transferred from the plasmid into the bacteria. If the gene markers are off, so if it does not have antibiotic resistance, or if there is no fluorescent protein flag on that cell, then no, that bacteria didn't allow that new plasmid in. Those are two ways that we can tell. Let's take a chance to go into detail on both of these. So antibiotic resistance here. The first way to identify a bacteria that contains, again, a recombinant plasmid, contains DNA from two different sources. You can actually grow those bacteria that you allowed in that little warm bath to let those plasmids inside of them, and only 0.001% actually got inside. You can grow whatever bacteria are resulting from that process on a growth medium. That just basically means Jello on a plate. And those bacteria will grow into colonies that look like circles. Usually these plates contain an antibiotic. Remember, plasmids contain antibiotic-resistant genes. And whatever you grow this bacteria on, in theory, it should, that's supposed to be a star, it should be resistant to whatever you're growing it on. Let's think about that before we move further. Our plasmids contain antibiotic-resistant genes. They might contain a gene here that says, let's resist penicillin. They might contain a gene here that says, let's be resistant to ampicillin. Let's say that you take some new DNA. Let's say this is your new DNA over here. So I'll put new. You get your restriction enzyme scissors out. You go cut, 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 and you cut a chunk out. You insert that new DNA in right there, and it looks like you cut the penicillin gene. If one of these growth plates contains penicillin, do you think that bacteria is going to be able to live and resist the penicillin? You're correct. No, it would end up dying because you broke that penicillin-resistant gene. However, if you don't, let me back up. You want to insert the new gene. Let's say you cut your plasmid over here. And then you insert that new gene here. That penicillin gene is still intact. That plasmid will still help the bacteria resist penicillin and it won't die. So over here, we can see there are a lot of colonies of bacteria growing. Over here, not very many, they were very small. So the bacteria on this plate, it means yes, they do contain the resistant genes, meaning you inserted the new gene in an area of the plasmid that still allowed it to have resistance. So those bacteria would be kept. This process is called replica plating. Replica plating, let me back up so we don't have too much stuff on the screen at once. So again, replica plating means you're growing groups of bacteria on several different plates. In that plasmid, there's multiple antibiotic-resistant genes. So you might grow that group of bacteria on a penicillin plate, an ampicillin plate, all sorts of different antibiotics. And depending on which one they actually live on, you're going to be able to easily see, wow, those bacteria are alive. They resist that. We inserted the gene correctly. So again, the new DNA should have been inserted into a gene that scientists know about. If it's in an unknown gene, scientists aren't going to know how to identify it. So if bacteria live, the recombinant plasmid was inserted correctly. Most important. And these bacteria would then actually be harvested by the scientists. They would take them off the plate and allow them to reproduce and reproduce to make whatever medicine they wanted, or whatever the gene was that they inserted into that plasmid. And again, if the bacteria dies, meaning this plate over here, where we'd see a couple tiny little colonies, but not much, you know that the DNA was not inserted correctly because those bacteria were no longer resistant. Hope that helps with our first type. Next slide is a picture of replica plating. So a scientist would basically take a stick with some cloth on the end. They would stamp it down on that plate, the original culture plate. So the original bacteria, which they allowed into that bath, some might have the plasmid, some might not. They're not sure which ones are which. They take it and they do a stamp of it. So these are identical right here. And then with this stamp, with all those multiple bacteria, they go stamp, stamp. They see what happens when they grow them in a normal growth gel, normal medium, or a medium growth gel containing certain antibiotics. This way, they can see, oh, normally, wow, a bunch of them grow, awesome. But when you add in antibiotics, that purple is really hard to tell. Let's pick yellow for our highlight color instead. When you grow antibiotics, when you grow them in antibiotics, look at that. Only a couple actually live. Those would be the bacteria that a scientist would say, okay, let's collect those and allow them to grow because these, check mark, have the new gene. They lived, we didn't cut an antibiotic resistant gene to get that new recombinant plasmid created. I hope that helps as we walk through the pictures, as I don't just show you a picture and say, this is it. When I actually draw on it, I know that, again, you guys are very visual, that it should help in understanding that process. Second type of gene marker was fluorescent markers. This one is very cool. You guys are going to love it. Here we go. So with fluorescent markers, we actually take a gene out of a jellyfish, which naturally produces a green fluorescent protein, or GFP. So GFP right there causes this jellyfish deep, deep in the ocean where there is no sunlight to be able to receive, or well, not receive, but actually make, out of protein, make its own light, which is super cool. Scientists use it all the time. They incorporate it into plasmids. This new DNA fragment, the GFP, the new DNA fragment that you want to insert into the plasmid, you attach it to the GFP gene. Let's see what it looks like. Here's our plasmid. Let's say, here's the new gene we want to insert. We would also attach the green, I'll use there, the green fluorescent protein gene to it. So as you can tell, we're actually inserting two genes rather than just one. With this green fluorescent gene, bacteria will glow if they contain that new gene, because we attached the GFP, or green fluorescent protein gene, to it. Super easy way to identify if something has a new gene. Take a look at these animals down here. They had new genes inserted into their cells with the GFP, green fluorescent protein gene, attached, and now they're glowing. Check it out. Rabbits, mice, chimpanzees, fish, worms, looks like a bee or some sort of bug. Definitely, all sorts of animals can easily be identified. Let's clear those annotations because there's a lot on the screen at once. So you could even use this if you're using bacteria to make antibiotics. You could grow them on the antibacterial gel plate, just like before with replica plating. And you could even attach a gene to an antibiotic-resistant gene so that you can look at those growth plates of bacteria under a light and see that they glow. That would be a way of easily identifying which bacteria contain the gene that you want. So again, if the DNA fragment has not been inserted correctly, then the bacteria would not glow and they wouldn't be used. This way is a very easy identifier and pretty fun. It's great to make things glow. All right, let's end with this Thinkpair Share. How is the DNA of a vector inserted into host cells? What are two gene markers and how do they work? Give 2 examples. Give you guys a few seconds. My neighbor texted me, so I'm going to see what's up with her real quick. Okay. She's just asking how everything is, which is great. I'm sure your neighbors are doing the same. So, I didn't have, let me go back. Previous slide. It's not letting me go back. There we go. Usually I have answers on these. I'm sorry I didn't. Let's write them. Pink for fun. How is the DNA of a vector inserted into a host cell? Vector was a word that we had talked about previously. Vector meant something that is used to transport. And in this case, the round plasmid in a bacteria was our vector. So how is the DNA of a vector, so the genes on a vector, inserted into a host cell? That was the process of putting a bacteria into a nice hot bath. And those bacteria, if their cell membrane becomes permeable, they allow that plasmid inside. They would actually take it in. Bacteria like having plasmids. They contain a lot of genes that are helpful, like antibiotic resistant. Why wouldn't that bacteria want that gene? Of course it would. So they allow plasmids inside. So that was the process. Do you remember how it was spelled? Transformation. Transformation was the process of how a DNA gene of a vector is allowed inside of a bacteria. That's how we're going to take that gene that we cut up on day 1. Day 2 attached it to a plasmid, and now we can allow it day 3 into a bacteria. Next question. What are gene markers? How do they work? Well, it says give 2 examples, so that gives us a hint. There are 2 types of gene markers. So what are they? First, we have antibiotic resistance, which was where in that plasmid they do have genes and sections that allow them to resist certain type of antibiotics. So if you add a brand new gene in, let's use blue, if you do not cut these antibiotic resistant genes and they stay intact, then check mark, you're good. That new gene can be identified by bacteria who live on those antibiotics because they contain the resistant genes. Our second example here, is GFP, green fluorescent protein, which allows the bacteria to glow. Who did we get it from? A jellyfish, because they naturally produce it. So if you attach that fluorescent gene to a bacteria, you'll easily be able to see whichever ones glow, they're going to contain that gene that you wanted. All right. I hope you all enjoyed day 3 of DNA technology, where we learned 2 steps today. Step 3, transformation. How do you actually get that bacteria to become recombinant or become a hybrid, taking in that plasmid who has extra genes? And also step 4, which was identification, and the 2 ways of visually identifying exactly which bacteria contain that new gene. Bacteria are tiny, you can't see them with your naked eye. So it's good to have a really big visual, like antibiotic resistance, they're growing or they're not growing, or green fluorescent protein, where they are literally glowing and you can see those cells. All right, I hope everyone is having a really great time. Please keep filling in your notes. Love you guys, and I'll see you tomorrow for, let's see, it'll be day 4 of the lecture and step 5.